Hello everyone. So I recently gave the interview for integrated PhD uh, at IISER Pune. So the interview was online. It was on 17th of March. The selection was based on the TIFR score which held in December. So on the basis of those score, students were selected, and I was invited for the first round of interview. And the date was on 17th of March. So there were four to five interviewers, and only two of them asked me questions. And the total interview, like it is mentioned in the mail, that the interview will last up for about 10 to 12 minutes. Mine lasted for about 11 minutes. So here are the questions which they asked me. First question, that why do you want to do integrated PhD at IISER Pune? And I said that it will provide me hands-on experience. They have good labs. I have interest in research, and a mixture of all these thoughts, and I gave the answer of it. The next question they asked me was about my internship. They, in a very detailed manner, said me to explain about my internship. I explained everything: what were the methods, what was the purpose, and what were the results. They asked me, this said me to re-explain a step. I explained it. Now the thing is that I did not revise much about my internship, because I heard from somewhere, or read online, that it's going to, the first round is very interdisciplinary. They're gonna ask you math and chemistry. So my focus was much on that part, and what I'm strong at or what I have done, I did not give priority to that. So I couldn't recall the specific methods and concentration of the chemicals, what kind of chemicals were used, and I couldn't answer that. Now this was a very very bad impression that went on them. It just made me look like that I haven't done the internship. So I got very very demotivated. My brain went into this defensive mode. Next, they asked me about what are the experiments I did in my bachelor's, and foolish me, I said, "So I haven't done any experiments. I haven't done any practicals." They were shocked that I said it was mostly coursework. Whereas I have done experiments in my college, in my internship, but I just couldn't say at that moment, because in my brain it was like, if I say something, they're gonna ask me a question, and what if I'm not able to answer it? So I just got very very demotivated then and there. Then they started asking me about PCR technique. I said about it. Then they asked me that what type of special polymerase is used. I said it's, yes, it is dark polymerase, where it is extracted from. I answered that. Then they asked me that why can't we use the normal human polymerase, DNA polymerase, which is present in human body. I said it is not heat stable. So he said that okay, we can increase the temperature, we can modify it, we can increase the temperature. Then why can't we use it? I said because it is competent to the environment present inside the human cell. Then he said, okay, we can provide the all those environment in the lab, and I couldn't answer it. Then this was the last question. The second last question which they asked me was about it was again from my internship. They, because there was gel electrophoresis done in my internship, they asked me that the results which I'm claiming in my internship. So they were asking me about negative control, about the ethanol and DNA interaction, and again I couldn't answer it, and it went very very horrible. And after this specific interview, I was just questioning my entire existence on this entire planet. I got very very demotivated, but days have been passed, and right now I'm okay. So here are my learnings which I have learned from this specific interview. Number one thing, not just skimming the surface, you really need to deep dive. And when they were asking me about the PCR technique, they already knew what my next answer is going to be. That means they are not looking for students who have superficial knowledge. And I was such a foolish person that what I'm really interested into, I did not study that. Also, they asked me about my topic of interest and subject of interest. I said it was genetics and molecular biology. Now, when they asked me questions about it, I wasn't able to, answer it. Next learning would be to have a really deep knowledge into one subject of interest. If you're in a bachelor's right now, or if you have pass out, please have one subject in which you are really really interested into. Please have a deep knowledge about it. You can search about it on YouTube, on AI, on internet, just anywhere. Have extra knowledge apart from the syllabus that is mentioned in the all these examinations. Second, please have one to two topic of interest, because they are going to ask you that what's your topic of interest, so you should be able to answer that. Have really deep knowledge about your internships and your BSc practicals. What all internships, what all techniques and tools were used during your course, during your internship, why that specific concentration was used, why the specific chemical is used. For example, tris and edt are very common chemicals which are being used. Now, why are they used, why are they specifically used in plasmid isolation? And last, I would say, please prepare any your subject of interest plus biochemistry, because come on, proteins is everywhere. At the end of the day, they're gonna ask you about proteins, cell signaling, and all these mechanism. And you should have clear cut knowledge about techniques. They're gonna give you situational base. They're gonna put you in this specific situation, and they're gonna ask you that why this band is not shown, what alternative techniques we can apply for a protein, RNA, DNA, or interaction, or any other molecule, any other specific chemical, protein, what all techniques can be used. So write down all the tools and techniques which you have performed during your bachelor's and during your internship. There are so many techniques mentioned in all these examination syllabus. Have a deep knowledge about them. Now, I'm going to use all these learning for my future, and I will make sure that I will not repeat all these points in future. And that's it about my traumatic experience with the IISER Pune interview. Thank you.